How amazing is life? You and I are made up of trillions and trillions and trillions of cells, even on the order of 325 trillion cells. And what's really amazing is all of those cells, despite having the amazing diversity of functions like heart cells, brain cells, eye cells, what have you, despite their diversity of function, they're all descended from one cell. You started off as one cell, one fertilized egg. And that fertilized egg divided into 2, and then 4, and then 8, 16, etc. And by the time you were born, there were trillions of them making you you. And that process has continued to this day and will continue as long as we live. And that process is called mitosis. So that's the topic of today's segment. We're going to learn about the life of a cell called the cell cycle and the way that those cells go about dividing to reproduce themselves. So all the cells in your body do this with the exception of your gametes, better known as your sex cells, your sperm or your eggs. With the exception of those, all the cells in our body undergo mitosis to reproduce themselves. So let's get started and learn about how this amazing process happens. It's simple yet elegant. So we can define mitosis as nuclear division of a cell producing 2 daughter cells. And the overall way it happens in animals like us is kind of like you were making some bread. You had a ball of dough and an unexpected guest came over. And so you took that ball of dough and you pinched it and you snapped it and boom, now you have 2 identical balls of dough by which you can make 2 identical loaves of bread. So very similar in animal cells, our cells are going to pinch and boom, now you have 2 identical baby daughter cells. So again, mitosis is going to happen in 4 phases: prophase, metaphase, anaphase, and telophase. So I want you to get out your fingers here and we're going to pretend our fingers are chromosomes. So you've already learned about what a chromosome is, what chromatin is, and what chromatids are. So let's get out our little chromosomes. We're going to say these are duplicated chromosomes and we're going to say prophase, do it with me, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. See if you can do it 5 times fast. So here are some beautiful pictures showing what the cells look like during each of these stages of mitosis. So here's prophase, metaphase, where the chromosomes are lined up down the middle, anaphase, and telophase. Now, before mitosis starts, and I'm going to move my head out of the way here so you can see. Before mitosis actually starts, it's in that gap 1, S, and gap 2 phase. And those 3 phases together are called interphase. So think of it like intermission. It's everything the cell's doing before it actually starts mitosis. And so during interphase, as you can see in this computer-generated model of a cell here, the chromosomes are in their chromatin format. So the DNA is just loose. It looks like a ball of string. You can see the nuclear envelope here. Lots of metabolic activity engaged in, that the cell's engaged in. You might be able to see the nucleolus. It's not shown in this picture, but here's the nucleus. Very visible. And you have your centrioles inside centrosomes here, right? So here are your 2 centrioles. They're visible. And they, right at the end of interphase, they're starting to separate from each other, but right now they are together. So here's a actual photograph of a cell that's been tagged with fluorescent protein. And there's the nucleus and you can see the rest of the cell. So just undergoing its normal growth and production of organelles and synthesizing its DNA, things like that are happening during interphase. So interphase, not technically part of mitosis. Now watch, I'm going to go back to interphase. I want you to watch the computer-generated model of the cell. Watch the centrioles, what happens. This is self-made animation here. Boop. See that? I'm going to go reverse again. Boop. Cool, huh? That's my animation abilities on PowerPoint. All right, so during prophase, so this is this part, right? I have you do your fingers like this because now the chromatin is going to start to condense into the chromosome format. And you've already learned how that happens with it wrapping around histones. So the chromosomes are now forming, they're condensing, they're the duplicated type, the X-shaped one. The nucleolus and the nuclear membrane are going to start to disappear. as you just saw, those centrioles are going to start to migrate to opposite sides of the cell, and they're going to start growing little spindle fibers made out of protein. And those are going to start extending as the process goes on. And the chromosomes are starting to shift. They're starting to migrate towards the center of the cell. Okay, so prophase. After that comes metaphase. So now you can see from our computer-generated model, those chromosomes have now lined up perfectly down the center of the cell. See? And that's called the metaphase plate, where they line up vertically down the center of the cell. Notice that the spindle fibers have now extended from the centrioles to the middle of each chromosome. And the middle of the chromosome, recall, is called the kinetochore. So the spindle fibers are attached there, and they're just lined up perfectly. So you can see in this real cell here, in blue, we show the chromosomes just lined up perfectly, and you can see the spindle fibers over here extending from the centrioles to the actual chromosomes. So duplicated chromosomes line up down the metaphase plate. Now watch this. Watch the computer-generated cell here. From metaphase, we go to anaphase. Did you see that? Let's go back. Metaphase, anaphase, metaphase, anaphase. So you see what's happened? We've now taken those duplicated chromosomes and split them down the middle. So now the separate sister chromatids are starting to be pulled to opposite sides of the cell. So look at that. They're being pulled over there, and you can see in the actual cell photograph here, see the chromatids, which are unduplicated chromosomes, are now being pulled towards each centriole. Now this is called anaphase. Ana means opposite. So a way you could think of it is Anna kicks her boyfriend out and they move to opposite sides of the town. Do that with me. Move to opposite sides of the town. So in other words, the sister chromatids separate, and those separated chromatids move to opposite sides of the cell. So far we have prophase, metaphase, anaphase, and now the next phase, you see what's happened? Back to anaphase, telophase, anaphase, telophase. So telo means end. This is the end phase. And so now those separated chromatids are now going to scrunch around each centriole, and they're going to start unwinding back into their chromatin form, their stringy form. So you can kind of see what's happening here, a set of chromosomes here, a set of chromosomes here. And now new nuclear envelopes, new nuclear membranes are going to form around each set of those sister chromatids. So we're creating a new nucleus on each side, and everything unwinds. And so each side now, each set of chromatin is identical to each other, but on opposite sides of the cell, which is now starting to pinch in the middle, because the next step, boom, is going to be cytokinesis. So cyto means cell, kinesis means movement. So now that after telophase, the cell in animal cells like ours is going to pinch just like the ball of dough and snap. And now we have two full cell membranes around each what was just the spindle fibers from, or the chromatids from before. So we have a new cell here, a new cell here, and now the nuclear envelope's already formed around this set of DNA, and nuclear envelope formed on that side of the DNA, and the centrioles are disappearing. Everything's going back to what the original mother cell looked like, but now times two, right, in two daughter cells. So these two daughter cells from one original mother cell that has split, we now have two identical daughter cells, and you want them to be identical, right? If you get a scratch on your arm, you want mitosis to produce new daughter cells to fill in that gap, and you want the skin cells to look exactly like the skin cells that they did originally, right? You don't want liver cells growing where your skin's supposed to be. So now for animals like us, we have that pinching off, and that results because you get a little ring forming in the middle called a furrow, and that will allow the whole cell to pinch off. Plants do something a little bit different. Plants will, because they can't pinch, they have a rigid cell wall. So instead, they're going to get little bubbles forming here, called, where you remember vesicles. The vesicles are going to start forming there with all the components needed to form the cell wall, and that will become a cell plate that will just extend out, and now you have a new completed cell wall. So now you have a cell wall around each of the new daughter cells.